Marcela is my wife. She is not feeling like presenting, so I'm going to do this for her. So we're going to talk about a project that Svetlana Lima and my wife, Marcela Bicalho, both jointly conducted in our lab. This was part of Dr. Lima's PhD thesis, and it's going to be published pretty soon in the journal PLOS ONE. So we're very interested in the microbiome, and for obvious reasons, right? So I think this illustrates a little bit about the general interest about the microbiome of humans and obviously the microbiome of animals. We're talking about 100 trillion symbiotic microbes live in every person, make up the human microbiota. There's about 152 genes from your microbiome compared to your own genes in your genome. And some interesting random facts here. There's about 2 kilograms of bugs in each one of us. There is a lot of new knowledge being generated about the microbiome. And this revolution on understanding of the microbiome and the correlation between your microbes and your own health, or the health of the host, was basically allowed by the same reasons that the previous speaker mentioned here, which is basically our ability to sequence DNA in a very effective and cheap, affordable way. For humans, it's unbelievable the amount of high-quality research published in very high-impact journals that have associated things like your microbiome and your gut health, your microbiome and your mental health, your microbiome and diabetes, obesity, and even your mood. So, and some of these research are worth taking a look at. So it's pretty fascinating. How can we look at the microbiome? Well, there are different techniques, and you can do what's affordable. And this is what we're going to focus on today, which is the sequencing of a part of the 16S ribosomal DNA gene. The 16S gene is an integral part of bacteria. And each bacteria will have 2 to 4, up to 6 copies of this gene. It has about 600 base pairs, and regions of this gene are very conserved and have very little variability between species of bacteria, between genera of bacteria, but other regions are what we call hypervariable regions, or basically regions that have an enormous amount of variation between different kinds of bacteria. And we focus on those regions. And the region that we focus currently is the V4 region. We don't need to sequence the entire 16S gene, so we just focus on that region. It's about 300 base pairs long, and it's incredibly variable between different types of bacteria. So it allows you to differentiate the different kinds of bacteria present in a sample in a very affordable way. When I say very affordable way, it is really affordable. So a sequencing kit of a MySeq sequencer from Illumina costs about $1,200, and I can run up to 300 samples. I can multiplex it to 300 samples. In theory, you could do more. You could do 1,000 samples in one run of a MySeq sequencer and still get good coverage depending on what you're talking about. A little bit more expensive is the metagenomics approach. In this case, you actually sequence the entire microbial DNA, not only the microbial DNA, but also the host DNA is there. So basically, it's shotgun sequencing. You take your sample, extract DNA from the sample, you cut it up, ligate it into your adapters, and then you run in your sequencer. And the advantage of doing that is actually you're not only going to get information on the what's in there, phylogenetic information, but you're also going to get information about what can the microbiome do, so functional potential. Interesting, but more expensive. And then we have, RNA-based technologies or techniques, and metaproteomics and metabolomics as well, all part of the deal here. Today we're going to be talking about the influence of the maternal microbiome on the neonate, on the health of the neonate. And I'd like to share, this is not from our lab, this is from Dr. Knight's group, but it's a fascinating little piece of work. And what you're going to see here on the very top is the microbiome of a child from fecal microbiome. You can see that this microbiome is, as the child ages, and here is the age of the child, up to 27 months. The microbiome of the neonate human resembles the microbiome of its mother's vagina. And it will continue to resemble vaginal microbiome for a very long period of time. And that is exactly because of vaginal delivery. That's your first microbiome, and that stays in your gut for a very long time. And here is the adult microbiome, and you can see that slowly, as it gets to 27 months of age, 2 years, finally, that little human being is going to have a microbiome that resembles an adult fecal microbiome. So we wanted to take a look at that in the cows, and we performed a study where we collected samples from 80 cows. We collected fecal samples from the cows, and we collected vaginal swab samples from the cows. When their babies were born, we collected samples at 3 days of life, at 14 days of life, and at 35 days of life. And here we have nasopharyngeal swabs and fecal samples. So we analyzed hundreds of samples in this study. So these are some of the questions that are interested in the analysis, looking at the beta diversity, alpha diversity, and taxonomic profile. And this is actually, it turned out that when we do this principal component analysis, this is a 3D graph. If we move it around, it would be very similar to the animation that I just showed you. But these are actually cow samples. So when you look at the calves, their feces is clustered with together here on this side, very different than the fecal material from their mother. So nothing, it doesn't resemble the adult fecal microbiome. But you can see that it evolves. This is the fecal samples at 3 days, and then at 14 days, and then at 35 days. And you can see that it is actually closer to the vaginal microbiome and the upper respiratory tract microbiome than it is from feces. It takes a while for that to go. But the remarkable thing here is that we also looked at the upper respiratory tract microbiome, and the upper respiratory tract microbiome of calves are quite similar to the vaginal microbiome of dams. So it's all mucosal tissue, and there's actually some microbes that are shared between those two environments there. We're going to detail them later on. So this is the phylogenetic classification. We have here the fecal samples from the mothers, from the dams, and this is very classic, where we have a division that is basically between Firmicutes, the gray bar, and Bacteroidetes, which is this part here. This happens on our own microbiome as well. It's basically dominated by these anaerobes classified as Firmicutes and Bacteroidetes. But you can see that it takes a while, and by 35 days, there's already a significant amount of dry matter intake consumption, but the fecal microbiome hasn't really caught up to what it should be. So there's still, it looks like what happens here is Bacteroidetes is underrepresented in the beginning, and it grows with age, and at some point we'll get to where it needs to be. When we look at the vaginal microbiome and the upper respiratory microbiome, there's incredible similarity. So this is Firmicutes and proteobacteria. The proteobacteria that is overrepresented in both microbiomes here is Mannheimia, Mycoplasma, Moraxella, and Pastorella. These are the major microbes. We're going to talk about that. There's incredible similarity between the vaginal microbiome of the mother and the upper respiratory tract microbiome of the calves, and that changes slightly over time. So this is basically a heat map showing the most common microbes. The dark part of the bar here is the higher relative abundance of the microbes, and the lighter is the low relative abundance. You can see the meiheimia, which is the primary cause of pneumonia. We're going to talk a little bit about that, is in low prevalence in the beginning of the life. This is the vaginal sample from the mother, and this is the upper respiratory tract sample from the calves at 3 days, 14 days, and 35 days. 14 days is when you start seeing pneumonia in these calves, and you can see the meiheimia is at very high prevalence at that point. And mycoplasma is here, it increases as calves start being diagnosed with pneumonia as well. Okay, and one interesting finding here, when we looked at the health of the newborn calves, and separated the calves based on the microbiome of their mother's vagina, we were able to find some differences between healthy calves and sick calves. And those differences are basically attributed to 4 microbes. And here we have, over to this side here, are the microbes that are overrepresented in healthy animals. And over to the left side, the microbes that are overrepresented in sick animals. So we find 2 microbes that, when present in the mother's vagina, was detrimental for the calf's health. One is Porphyromonas, and the other one is Campylobacter. Interestingly, meiheimia, which is the primary cause of pneumonia, was actually preventative when present in the vagina of their mother. So cows that had high counts of meiheimia in the vagina, their calves were more healthy. So that could be a probiotic effect, that could be a vaccination effect, or a combination of both. We don't know, but it's very counterintuitive. When we look at the dynamic of the microbiome of the calves, and we look at birth, 3 days, 14 days, 28 days, and 35 days, we found some interesting results. First of all, we are able to identify basically all major bacterial pathogens in all calves at day 3 of life. Meiheimia is there, Mycoplasma is there, Moraxella is there, Pasteurella is there. So you name it, those pathogens are going to be there in the upper respiratory tract right after birth. Now the interesting thing is, Meiheimia is present in 100% of the calves, but in very low quantities. You can see as pneumonia starts to be diagnosed, day 14 to 20 days of life, this is classic in zootic pneumonia, you start seeing that the relative abundance of meiheimia really shoots up in animals that are diagnosed with pneumonia compared to healthy animals. So pneumonia animals have a blooming of meiheimia in their upper respiratory tract, and this is probably the primary cause of pneumonia. Otitis as well, and a combination of pneumonia and otitis as well. Mycoplasma, as you know, classic microbiology studies have demonstrated, it lags. You know, the primary pathogen is probably meiheimia, but you can see that after meiheimia peaks, Mycoplasma kind of shows up, and it's like they join the party as an opportunistic pathogen. And you can see that the calves that were diagnosed with pneumonia had twice more Mycoplasma microbes in relative abundance compared to the healthy calves. And it was also very relevant for otitis and so on. What else? This is an unexpected finding as well. We found that the genus Moraxella was also associated with respiratory disease, both with otitis and pneumonia. Moraxella is a common cause of pink eye, Moraxella bovis, but we don't think that this is Moraxella bovis. We actually think that this is Moraxella cataralis, which is a common cause of upper respiratory tract infection in calves and also in children. The same microbe causes lots of mucus and irritation of the upper respiratory tract. So these probably needs to be further investigated to better understand it. And the curious thing here on this finding of this study is that Pasteurella was not associated with pneumonia at all. Actually, when you look at pneumonia in healthy calves, Pasteurella was more present in healthy animals than in pneumonia calves. So the microbe that we have been blaming, Pasteurella mutacida, as the primary cause of pneumonia, in this case, has shown actually to be protective.
You need to wrap up, please, soon.
Yep, so this is the last slide, conclusion, and that's it. Thank you.